And the other technique that we can use is called affinity chromatography. Affinity chromatography. And in affinity chromatography, let me tell you, affinity chromatography is incredibly powerful. Really, really powerful. It's so powerful that you can sometimes purify your protein in one step. Okay? But it requires two things. It requires that you know a ligand of your protein. That is, you have to know your protein wants to bind to something. And it requires that you can make a column with the ligand attached. And the idea is really simple. What if I want to purify a protein that I happen to know binds to glucose? Okay, I could make a bead, and on top of that bead, I could stick a bunch of glucose. Important. G here is glucose. It is not glycine. I'll say it again. G here is glucose, not glycine. And if I have a protein that likes to bind to glucose because it's maybe got a glucose binding pocket, here's my protein. I can purify this away from everything else by just taking my crude cell goo and putting it on this bead right like that. Okay? That will work fantastically well. Now, how do I get it off? If it's stuck to my bead, it's glucose binding, it's stuck to the glucose on my bead. How am I going to get it off the bead? What could I add to compete? Glucose. All I got to do is add some glucose, and that'll compete it off. Okay. Now, two examples of this. Okay, suppose I wanted to purify transcription factors. What might I want to put on the bead? Transcription factors are proteins that bind to specific DNA sequences. What should I put on the bead? DNA sequences. Okay? Now, you can suppose I want to purify a protein, but I don't know what it binds to. Could I engineer it to bind to something? So could I use genetic engineering? Engineering a ligand. Could I use cloning and genetic engineering to stick some kind of a tag onto it? Sure, what could I use? I could use a his tag, okay? A histidine tag. I could take the DNA from my protein, and I could stick a bunch of codons, we'll go through this at some point, for histidine, so that my protein, which ordinarily would be like this, now has stuck to one end of it a whole bunch of histidine residues, H, 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 H. Okay? And it turns out that histidine has a very high affinity for certain metals, like nickel. Cobalt. So I could make a nickel column, and I could add histidine. Okay? I'm sorry, I could express this protein. I've engineered this. I could express this in some other organism, like bacteria, and then I could use a nickel column to purify it off. What could I use to get it off my nickel column? Histidine, or what's the side chain of histidine? Imidazole. I could just use imidazole. Anybody know another kind of tag? Who's ever heard of GST? GST stands for glutathione S-transferase. This is a protein, a small protein, that I could stick, not so small, about 26,000 Daltons. I could stick it on the end of my protein, and now my protein will bind to glutathione. So I could make a column with glutathione on it, and my protein would stick to glutathione.